Welcome to this explainer. Today we're going to take a look at something we all carry around inside us every single day. It's absolutely vital for our survival, but completely invisible to the naked eye. We're going to dive deep and explore the beautiful, hidden 3D structures that physically define our blood types.
We hear these letters all the time, right? Type A, type B, type O. But what exactly is the difference between them? It's not just some random category on a medical chart. If you were to literally shrink down to a microscopic size, what physical differences would you actually see on the surface of a blood cell? Well, today, we're going to go way beyond just letters on a page and uncover the microscopic architectural differences that literally make up who we are.
So, here's our agenda for today. First, we'll cover the discovery of ABO. Second, the genetic blueprint. Third, the 3D molecular building blocks of our blood. Fourth, Landsteiner's law in action. And finally, the strict rules of transfusion. Let's get right into it. Section 1, the discovery of ABO. Let's rewind the clock for a second, back to the early 20th century. The year was 1901, and a scientist named Carl Landsteiner made a truly groundbreaking discovery. Up until this point, blood was kind of just thought of as, well, blood. But Landsteiner figured out that there were actually distinct antigenic differences between individuals of the exact same species. What Landsteiner actually showed was that we have these naturally occurring antibodies floating around in our blood. He called them isoagglutinins. He noticed that if you mix the blood of certain people together, these isoagglutinins would react with and aggressively clump together, or agglutinate, the red cells of other individuals. And boom, through this reaction, he discovered the ABO blood group system. And honestly, it remains easily the single most important system in all of transfusion science today. Moving on to Section 2, the genetic blueprint. So you might be wondering, how do you actually end up with one blood type over another? Well, the inheritance of the ABO genes follows your classic, simple Mendelian principles. The O gene is recessive, while the A and B genes are codominant. Because of this recessive nature, having group O blood is only seen in people who are homozygous OO, meaning they inherited an O gene from both parents. It's really that straightforward. Okay, let's dive a little deeper here because this is where it gets super cool. Your genes don't actually code directly for the blood type antigens themselves. Instead, they code for enzyme activities called transferases. Think of transferases like a tiny crew of microscopic construction workers. Their one and only job is to pick up very specific sugar residues and attach them onto a pre-existing precursor molecule that just sits there on the surface of your red blood cells. Section 3, 3D molecular building blocks. All right, let's zoom all the way in and look at this 3D construction sugar by sugar. It all starts with a foundation. Imagine a tiny chain protruding right off the red blood cell. First, you've got a base of glucose. Snap on a galactose, then N-A-glucosamine, and finally, another galactose. This specific chain is the basic precursor substance. And the crazy thing, it stays exactly the same for everyone, totally regardless of your blood group. It's the universal foundation. Which brings us to the next absolutely crucial step, the creation of the H antigen. The H gene codes for a specific transferase, alpha-L-fucosyl-transferase, which basically grabs a molecule called L-fucose and sticks it to the very end of that precursor chain we just talked about. This new structure, that's the H antigen. Now, if you're group O, your genes actually don't code for any additional functional transferases. The O gene is essentially an amorph, like a silent gene that doesn't produce a working construction worker. So for all you group O individuals out there, construction stops right here. The H antigen is your final product. Now, here is where the path splits, and it's fascinating. If you have the A gene, your transferase adds a very specific sugar called N-acetylgalactosamine right to the terminal sugar of the H antigen. But if you have the B gene, a completely different transferase steps in and adds D-galactose to that exact same spot. Think about that for a second. It's literally just one single sugar molecule, making the entire difference between type A and type B blood. Mind-blowing, right? Just picture these structures side by side for a moment. You have the H antigen structure for group O, then the A antigen with its unique N-acetylgalactosamine tip, and finally the B antigen tipped with D-galactose. These tiny, highly specific sugar residue variations physically alter the 3D surface of the red blood cell. Guys, this is the literal physical architecture of your blood. Section 4, Landsteiner's law in action. So, zooming back out for a second, how does the immune system actually respond to these beautiful little structures?
Well, it all comes down to Landsteiner's law. It states whenever an antigen is not present on the red cells, the corresponding antibody is found in the serum. In simple terms, your body naturally creates defensive antibodies against the 3D antigen shapes it lacks. So, if you have type A blood, you completely lack that B sugar structure, the D-galactose. Because of that, your body naturally produces antibodies against it to protect itself. And here is a highly fascinating fact about Group O individuals. Because their red cells lack both the A and B specific sugars, you might logically assume they just produce two totally separate antibodies, right? An anti-A and an anti-B. But actually, no. They produce a unique single antibody specificity called anti-AB. This single, highly versatile antibody is capable of recognizing and reacting with either the A or the B antigen. How cool is that?
Section 5. The rules of transfusion.
So how does all this biological architecture actually dictate the life or death rules of medicine? Well, before anyone can safely receive a blood transfusion, laboratories have to perform rigorous pre-transfusion testing using a really robust double-checking system. First up is the cell group, which tests the patient's red cells against known antisera to directly detect which 3D sugar structures, the antigens, are physically present. Second is the reverse, or serum group, which tests the patient's plasma with known red cells to confirm which isoagglutinins, the antibodies they naturally possess. By using these two methods together, medical professionals create a foolproof safety net to avoid any catastrophic incompatibility errors. And that brings us to the final picture of blood compatibility. Now, looking at this matrix, it might just look like a giant grid of yeses and nos. But armed with the knowledge of those 3D sugars and Landsteiner's law, it makes perfect logical sense. Group O can donate to absolutely anyone because they lack the A and B sugars that trigger an immune attack, but they can only receive from Group O because they carry that powerful anti-AOB antibody that is ready to attack any foreign shapes. We've traveled all the way from a historic discovery in 1901 right down to the tiny molecular construction workers attaching specific sugars to the surface of your cells. It's an absolute marvel of biological engineering. So I'll leave you with this thought. Next time you roll up your sleeve to donate or receive blood, will you picture the specific microscopic 3D sugars you're sharing? Thank you so much for joining me on this explainer, and keep marveling at the amazing structures hidden right inside you.